I want to move on and introduce our next speaker. Our next speaker is Dr. Deidre Harmon. Dr. Harmon is the Mountain Livestock Specialist for NC Cooperative Extension. She's stationed at the Mountain Research Center in Waynesville, North Carolina. Her program focuses on the utilization of byproduct feeds, especially wet brewers grains, and understanding how forage species, variety, and management practices influence the yield and forage quality. Recent studies have compared all available commercial varieties of novel endophyte tall fescue at 3 locations across the state of North Carolina, and how a nurse crop of oats or wheat influences the establishment of tall fescue and orchard grass. So with that, I'll turn it over to you, Dr. Harmon.
All right. Thank you. Thank you, Paul. And I'll go ahead and share my screen. Perfect. So this talk I usually give in person, where we all get our hands on some plants and really dig in and dive deep into what's going on with these plants. So I'm going to have a mixture today of doing some PowerPoint slides, but then I'm going to exit out of the PowerPoint and pull up some plants and show you some different things, parts of each plant. So just bear with me. And if you can't see what I'm doing, just let me know and I'll adjust. So today I have the challenge of talking to you about understanding plant growth and physiology. And really, if we think about plants on our farms, this is truly how we can think about utilizing those animals to graze our forages. We got a couple things to think about when we're talking about forages. Animals aren't necessarily like us. Animals don't graze or select plants to live. They don't select plants to protect their future meals. Now, I'm in the afternoon, I might make a meal and I hate cooking. So I'm going to figure out some way to make it last for the next 4 or 5 days. So I'm truly trying to protect my future meals, whereas animals really don't do that. They're just grazing to fill their bellies up. So they don't know anything about plant survival. And so as managers, our goal is to really think about, how we can control those animals to harvest that grass so that we protect that plant so it can survive for many years to come. And so as managers, we got to think about how close and quickly we're grazing to the ground. We got to think about, how long we're going to graze that pasture or that field, and then how much rest it needs so that we can regraze it in the future. We need to think about when and how to fertilize, and maybe if you're in an area of the United States where you have to apply water, maybe the timing and the application of that water as well. And so Dr. Poor's already talked a little bit about these things, but thinking about our control tools of fence, water distribution, and maybe even attitude and patience, right, on these things is really what we can do as managers to help control these plants to make sure that they can survive themselves. So overall, kind of what I'm going to be going into this afternoon is thinking about how we can better understand plant physiology and plant morphology, and how we can use those two things to really manipulate the factors which encourage rapid plant growth and survival, because we want those plants to survive and not have to go and establish new pastures or new hayfields every couple of years. So if we think about plant physiology and morphology, they're really talking about 2 different things within that plant. So morphology really describes the physical structures or the outward appearance or those different parts of that plant, whereas physiology really describes the chemical processes that are going on in that plant, the organic functions that allow it to grow and store carbohydrates. It's really the basis of how plants live, grow, and age in response to environment and management and the things that we do as managers. So I'm going to talk a little bit about how plant growth aspects will help answer a couple of different questions today. And we're going to go through a little bit of each one of these, but how, understanding plant morphology and physiology can help us understand how often we cut or graze those plants. Each species and each species of plant or grass or clover is really different. So we need to think about that. When to fertilize and how much, we got to think about, as Dr. Poor has already stated, the growing season, when to fertilize. Think about maybe your pasture, and I'll give you an example of this. If you have tall fescue, that's a pretty thin stand. How can we get it to tiller more? Maybe it's a good plan is to add nitrogen at the right time to allow that plant to really tiller, to increase our not only our cover of soil with forage and green leafy tissue, but also increase our yields and our quality of forage as well. Planting rates, dates, and methods are also something to think about. And I won't go in a lot into that today, but how persistent is it? An annual forage? Is it a perennial forage? We may manage our annuals quite differently than we manage our perennial forages. Tolerance to the grazing animal. Dr. Poor's already talked about the differences between maybe a beef cow or sheep, goat, and horses that can really be very selective in how they're grazing and graze much closer to the ground than a cow can. Also, landscape adaptation. We know that some plants tolerate more acidic soils better than other plants. We also know that some forages can tolerate more compaction than other forages, soil compaction. So it's really a ecosystem. And if we just get out there and start observing what's going on, that ecosystem tells us a lot about how we're doing as a manager and what we can do to better our management. So first off, we're going to talk a little bit about understanding different plant parts. And this is really key to understanding how plants grow and kind of how we can manage it from an animal perspective. So on the screen, the first thing, just kind of starting at the ground on this grass plant. At the ground area down here, we have the crown. This is really kind of the primary area of cell division, elongation. And this is where that tiller or that plant stem is really going to differentiate between a leaf and between maybe the inflorescence part at the top of the plant. And so this is really the area we get a lot of cell division and growth. Oftentimes, if you start digging down into the canopy, this area is on that green plant looks almost white or milky like. That's how we know that area is really that cell division area. As we start moving up, you can see on the screen that we've got a few different tillers or stems, if you will. We've got a really young vegetative tiller here that's young, very immature, that's just starting to come on. As we move up to this big tiller, we've got this big tiller that's put on a seed head. If you think about tall fescue around May time, where we've got tons of seed heads out there. Keep on moving up this plant. We really get into the sheath leaf collar area. We have a node here. The node is also a primary site of cell division, elongation, and growth, similar to that merry-stem down here at the bottom. And so right above that node, we have the leaf sheath. And that leaf sheath, it's photosynthetic, it's green, but its primary job is to really provide some structure to that leaf to allow it to hold it up. It also allows for nutrients coming up out of that leaf, out of those roots to go into that leaf. And so then moving up to that collar region, we have auricles, different parts that I think I may have a picture of later that's closer into that. And then once we get that stem cell elongation, we get inflorescence where we put on a seed head. So if we think about, I'm sorry, let me go back. And so there's some plants that may have a couple of other different parts. A rhizome here on the lower left side of the picture is basically a shoot that comes underground and then will start to come up out of the ground and create basically a new plant from that rhizome. We also have stolons that run along the ground. If you think of something like Bermuda grass or white clover, both of those species have stolons that run along the ground. At each one of the nodes on that stolon, that can actually root into the ground and produce new shoots. And so I'm going to kind of end my PowerPoint here and show you just some different plants that I have here sitting next to me. I think I can end it. Can you see me?
Yeah, if you just
Oh, I see it.
maybe stop share.
Okay.
Yep.
It was on my other screen.
There you
Okay.
go.
So just thinking about some of those parts that we talked about, stolons and rhizomes. I've got a couple of plants here that I can show you. So if we think about white clover, and I went and dug some white clover up today, and I'm in the mountains, so it's still pretty cold here, so we don't have a whole lot of forage growth here at this time. But you can see that this is a white clover plant. And this stolon runs on top of the ground, this part here. And so you can almost see that at each one of the nodes on this white clover, we've got a couple of roots starting to form right here. You see that? And then we've got leaves coming up out of those different nodes. So the growing point on this white clover is here on the very tip, the very end. So this is going to keep creeping along the ground and growing. And you can see another root right there that's starting to appear. So that's white clover. I've also got a fescue plant here. And so if you think about this fescue plant, we've got 4 different tillers coming out of this one plant. So 1 tiller, 2 tillers, a very young little tiller here, 3, and then another pretty young tiller here, 4. If we start going up this tiller, we can see that we've got the collar area on this plant, and I'm sorry if you can't see this very well. That's right here. If we pull this back, we can see that there's small little auricles that kind of wrap around this plant in this collar region. I don't know if you can see that or not. In a tall fescue, we've got a pretty strong mid vein in the leaf here. When we start thinking about identifying these different plant species, this collar region where this leaf comes out is really where we can help us understand what species it is. Each one of these different forage species has a somewhat of a different collar region. So by looking at this collar region here, we can help identify the different species that we have. I also have a pretty big red clover plant. And so the unique thing about this red clover plant is that if you look closely, you can see a couple of rhizomes starting to form here on this red clover plant. And so these rhizomes are going to shoot up and form new plants from underneath the ground. Okay, I'm gonna go back to my slide point now, my presentation now that I've got dirt all over my desk. Okay, can you see it?
Yep, you're good.
Are you seeing the right screen?
Yeah, the understanding plant parts. But we do have your slide notes.
Okay, let me switch.
Switch to your other screen. There you
There
go.
you go.
That looks good.
Okay, perfect. So going to the next slide, if we think about, these different plant parts and where the growing point of each one is, I've already talked a little bit about the growing point being down here in the meristem region. And as we move up, that plant where we have that ligule, that collar region right below that leaf sheath is where we have a node, and that's also the growing region. Where we have a collar present on that plant part, if we come in and we graze that off, we won't have a lot of regrowth there because we've basically cut off that growing point. So let's talk about the stages of plant development here and why that's important to understand. So if we think about that meristem or that shoot apex, which we talked about was in that lower part of that plant, where that plant gets pretty white if you start looking at a plant. If you see these different nodes here way down deep, you can see that we've basically got 1, 2, 3, 4, 5, 6 nodes, which means we can have 6 leaves coming out of this shoot here. As that plant starts to grow and these nodes start to push their way up into a tiller, producing leaves from each node, we start getting a reproductive tiller. And of course, this all depends on the environment, the length of daylight, depends on when this is going to happen. So if we think about fescue plants, we know that there's a lot of reproductive tillers that are starting to shoot up now. I've got one in my hand here that I just showed you where we had those 4 different tillers, and we've got that one pretty big reproductive tiller that you could see. And so once we get that reproductive tiller up, we get that flag leaf at the very top of that plant. We know that that inflorescence or that seed head is starting to push its way up through that leaf and will eventually emerge. And we call that the boot stage. And if we think about how quality and yield of forage kind of intersect, this is really where we are getting the most bang for our buck, if you will, where we've got the best forage quality and the best yield, if that makes sense. So once that inflorescence pushes its way up through that flag leaf, obviously then we've got the mature seed head. And if we go and cut that mature seed head, those tillers won't regrow. That's not where regrowth is coming from. We have new tillers that plant will start developing and growing, and that's where we get our vegetative plants at. If we think about clover or maybe alfalfa, it's same principles, but it looks a little different, right? So we've got the white clover here growing on the ground. We've got the stolon creeping its way. We've got the growing point here on the end. So this will keep pushing its way this way and producing new leaves. And alfalfa is a very similar situation where we've got different nodes, as the picture shows here, and producing different tillers from that root or that meristem at the ground level. So why does this matter when we start thinking about grazing height and how are we going to grow these plants? Well, if we think about that growing point and where that growing point's at, if we go in here and we graze this white clover at 2 inches, we never actually hit or graze off that growing point of that white clover. However, if we go in and graze at 2 inches, a plant like tall fescue, where we have that growing point elevated in that plant, it's starting to produce that reproductive tiller. That reproductive tiller is coming up and that inflorescence is coming out. Well, then all of a sudden we have hit the growing point and that tiller will not come back. So when we start thinking about this from a grazing point, and I'm going to end my slideshow again and get my plants back on here. We've got to think about where the storage organs are at in this plant. We've got to think about where the leaf material's at in this plant and the growing points at in this plant. So if we come in here to this white clover and we graze it down to 2 inches, we didn't necessarily hurt this plant at all, right? We didn't get into the carbohydrate storage of this plant, which is the stolon that sits right on top of the ground. We didn't get into the growing point of this plant, which is again right on top of the ground. And so this plant is going to continue rocking and rolling. We left some green material to photosynthesize, to catch the sunlight, to continue to produce carbohydrates for the plant. However, if I took an orchard grass plant, and you can kind of see where that white area of this orchard grass plant is, that's the growing point there. And we came in and we grazed that down to 2 inches. Well, what have we done now? One, we've taken all the green tissue off of that plant. So the only place that it can regrow from is to use its root reserves, because there's not a lot of sunlight getting on that green tissue because there's no green tissue left. And so it's having to use its reserves in the roots, the carbohydrate reserves in the stem here, to regrow new tissue. Now, once it regrows new tissue, the leaves can capture sunlight. They can start putting energy reserves back into the roots, and this plant just continues to grow. However, if we come in here and we consistently graze this plant down to 2 inches, it barely starts to regrow. We graze it again, it starts to regrow. We graze it again. Well, then now what we've done is that every time it starts to put a little bit of green tissue out, we go back and cut off that solar panel, if you will. And so the danger of repeatedly overgrazing these different species, such as orchard grass, is we never allow that plant to recover and rebuild its carbohydrate storage down deep in the roots and in the lower part of the plant. So that means that by continually overgrazing this plant, we can actually damage its reserves and end up killing that plant. And so if we kill those plants by overgrazing it, nature in general doesn't like bare soil. So there is some type of species that's going to come in and fill in that bare soil. Probably more than likely it's going to be weeds. So one thing that oftentimes we notice in overgrazed pastures is that we see a lot of clover in overgrazed pastures, specifically small Dutch white clover. And the reason that we see a lot of small Dutch white clover in pastures is because it can withstand overgrazing much better than some of our other grass species like orchard grass or tall fescue, because of where that plant keeps its root reserves, or its, carbohydrate reserves, which is close to the ground. We're laying right on top of the ground in that stolon. And so it can withstand a lot more overgrazing and pressure. And so if you go into an overgrazed pasture, you're probably going to see a lot of that small white Dutch clover in it, in that pasture, because it can thrive in that setting, whereas orchard grass and fescue cannot. Okay, let me see if I can share my screen again. Okay, so if we think about alfalfa and some of the particular plant or parts of alfalfa, I really like this picture because we can see the crown of the alfalfa plant. We've got new tiller shoots that are starting to emerge from that crown. This old tiller here would have been where they came in and maybe cut this forage or this alfalfa for hay. And so we've cut off that growing point. So this tiller is going to die. But all these new little tillers are going to come and take its place. And if you've ever looked at the crown of an alfalfa plant, you can see all those old tillers and those new tillers coming in to take their place. And I've got a tall fescue plant here that I dug up from a hayfield. And it's really interesting looking down into the crown of that plant because you can see all kinds of basically dead tillers that looks exactly like this alfalfa old cut tiller. And they're kind of sharp. If you kind of put your hand down on it, they're kind of prickly and sharp where they've been mowed and they're dead and they're nothing but dead tissue there. So my red clover plant, I really like it also because you can see in this plant the new tiller and shoots that are starting to emerge. We've got a lot of old tillers here in this picture, and red clover and white clover have two very different growing patterns. If you notice in this red clover, it's got more of a crown like alfalfa does, where it shoots up old tillers from the crown, whereas white clover, again, has those stolons that creep on the ground that will shoot up tillers from that stolon. So thinking about tall fescue and looking at it, tall fescue is pretty interesting that it actually has rhizomes. But the rhizomes and tall fescue are extremely small, and it's hard to really see them. This picture shows the rhizomes here at the very base of this plant. And so if we think about in this picture here where the region of cell division and growth is again, if you look down into that canopy of that plant right on top of the ground or at the ground level, that's really where that elongation is happening and that cell division is happening in that plant. If you have Timothy, we've got a similar story as that it has these live tillers and shoots coming out of that base of that plant. But Timothy's pretty interesting, as well as orchard grass, which I think I've got on the next picture, is that it actually has these little corms, which is basically a carbohydrate storage for that plant. And so if you kind of dig up an orchard grass plant and start pulling those tillers apart, you'll eventually get to a corm where those tillers kind of are attached to. And I actually have one of those. Again, I'm going to keep sharing and unsharing my screen. And so I actually found one of those while I was working on my orchard grass a minute ago. And so if you notice, here's a corm. I cut it in half with a razor blade, and you can see what a carbohydrate storage that is. And I know I'm already almost out of time, Paul, so I'm going to wrap it up here. So lastly, I'm going back and share this. So basically, we just have got to think about swap displays. Think about where those plant parts are and how we can utilize that to make sure that we are being the best managers possible. Now, we all know that the raw materials to basically photosynthesis is carbon dioxide from the environment, water, and sunlight. And we need all that in order to grow green tissue and green growth. So I'm going to skip these slides because I'm already out of time. So when we think about the energy reserves and storage organs of different species, and it may be different from your farm to the next, we don't have Bermuda grass up here, but Bermuda grass acts a lot like white clover if you're in a Bermuda grass area. If we think about where this carbohydrate or energy reserves are for Bahia grass, bluegrass, crabgrass, fescue, pearl millet, orchard grass, those different kind of species, it's really in that stem base, that lower 1 to 2 inches of that stem base. So we've got to make sure as managers, we're not allowing our animals to overgraze into that carbohydrate storage where that's at in the plants. For alfalfa, some of our clovers and other legumes, it may be in the tap roots. For like white clover, it's in the stolons. And then we've got a lot of different species that keep energy or carbohydrates or sugars, if you will, in the rhizomes. One of the major plants that I think about having rhizomes is Johnson grass. If you've ever tried to go dig up Johnson grass, you'll notice that it's got rhizomes going everywhere under that ground, shooting up new plants out of that forage. So lastly, I'll just leave you with this. If you're thinking about grazing and thinking about the forage species you have out there, think about where you can begin grazing and end grazing, specifically where you want to stop grazing at. For alfalfa, you can graze down to about 2 to 4 inches and still be okay. For white clover, you can really overgraze white clover and it's still going to survive and persist down to about 1 to 3 inches. If you have orchard grass is a little bit more delicate. We've got to make sure that we stop grazing at 3 to 6 inches. And then tall fescue, we can graze lower than orchard grass down to 2 to 3 inches. So with that, I'm going to leave it at that. And if you have any questions, I guess, take them at the end.
Thanks again, Deidre, for again another excellent presentation and just helping us understand. If we understand the morphology, the physiology of these plants, then we know how to graze them to promote survivability.